A plasmid preparation is a method used to extract and purify plasmid DNA. Many methods have been developed to purify plasmid DNA from bacteria. These methods invariably involve three steps: growth of the bacterial culture, harvesting and lysis of the bacteria, purification of plasmid DNA. Growth of the bacterial culture. Plasmids are almost always purified from liquid bacteria cultures, usually E. coli, which have been transformed and isolated. Virtually all plasmid vectors in common use encode one or more antibiotic resistance genes as a selectable marker, for example a gene encoding ampicillin or kanamycin resistance, which allows bacteria that have been successfully transformed to multiply uninhibited. Bacteria that have not taken up the plasmid vector are assumed to lack the resistance gene, and thus only colonies representing successful transformations are expected to grow. Bacteria are grown under favorable conditions. It is important to use proper controls to make sure that the antibiotic is actually killing bacteria that have not taken up the plasmid. This can be accomplished by plating non-transformed cells on a plate with antibiotics. No growth is expected on that plate. Other systems of controls can also be helpful. Harvesting and lysis of the bacteria. When bacteria are lysed under alkaline conditions, both chromosomal DNA and protein are denatured. The plasmid DNA, however, remains stable. Some scientists reduce the concentration of NaOH to 0.1 m in order to reduce the occurrence of ssDNA. After the addition of acetate-containing neutralization buffer, the large and less supercoiled chromosomal DNA and proteins precipitate, but the small bacterial DNA plasmids stay in solution. Preparations by size. Kits are available from varying manufacturers to purify plasmid DNA, which are named by size of bacterial culture and corresponding plasmid yield. In increasing order, these are the miniprep, midiprep, maxiprep, megaprep, and gigaprep. The plasmid DNA yield will vary depending on the plasmid copy number, type and size, the bacterial strain, the growth conditions, and the kit. Manip preparation of plasmid DNA is a rapid, small-scale isolation of plasmid DNA from bacteria. It is based on the alkaline lysis method invented by the researchers Baum and Doley in 1979. The extracted plasmid DNA resulting from performing a miniprep is itself often called a miniprep. Minipreps are used in the process of molecular cloning to analyze bacterial clones. A typical plasmid DNA yield of a miniprep is 50 to 100 microgram depending on the cell strain. Miniprep of large number of plasmids can also be done conveniently by on-filter paper lysing. The elution of the filter paper that contains plasmid can be directly sequenced to produce more than 700 BP high-quality sequencing data with CE sequencing. Midip preparation the starting E. coli culture volume is 15 to 25 milliliters of lysogeny broth and the expected DNA yield is 100 to 350 microgram. Maxship preparation the starting E. coli culture volume is 100 to 200 milliliters of LB and the expected DNA yield is 500 to 850 microgram. Megapreparation the starting E. coli culture volume is 500 milliliters euro 2.5 L of LB and the expected DNA yield is 1.5 to 2.5 milligrams. Gigapreparation the starting E. coli culture volume is 2.5-5 L of LB and the expected DNA yield is 7.5 euro 10 milligrams. Purification of plasmid DNA, addition of phenyl chloroform can dissolve and denature proteins, like DNase. This is especially important if the plasmids are to be used for enzyme digestion. Otherwise, smearing may occur in enzyme-restricted form of plasmid DNA. References: Baum H.C., Doley J. A rapid alkaline extraction procedure for screening recombinant plasmid DNA. Nucleic acids Res 7, 1513 euro 23 doi, 10.10937.6.1513. PMC 342324. PMID 388356. External links: http www protocol online org molecular biology maniprep / a maniprep procedure using diatomaceous earth to bind DNA during purification and washing.